Okay, I think we're ready to go. Sorry, we're just running a few minutes late, everybody. We've still got people coming in. We're sort of admitting them. We've got about 300 people joining this presentation today. So while they're still starting to come in, I'll just go through some housekeeping and talk quite slowly to give them more chance to join. So I should let you all know that the meeting is being recorded, and we will share a link to that recording, hopefully by tomorrow morning. So you should receive an email for that. And that'll also shortly be available on our website, along with a summary of the questions and answers that we'll be going through towards the end of the presentation. And the way we're going to go is that I'm going to talk for about half an hour. I think I've only got about 20 or 22 slides to go through. And there is a question and answer box that you should see. Please feel free to type any questions into that, either through the presentation or towards the end of the presentation. And then we might take a 5-minute break or so when I've finished going through my slides to enable us to just triage those questions a little bit and come back after about 5 or so minutes. And hopefully that'll enable us to address the questions as effectively as we can. Only we can see those questions, so don't worry about asking a question and anybody else seeing it. We will be addressing those questions anonymously. But if you would like to post your question anonymously, then you have that option as well. So that just leaves me actually to thank everybody for registering and listening to this presentation. So 7 o'clock on a Tuesday evening. I'm sure many of you have got things that you would rather be doing with your Tuesday evenings. I think we have people on the other side of the Atlantic, so it's a little bit earlier in the day for you. And I think we've actually got people tuning in from Australia as well, so it's probably early tomorrow. But I've given a lot of presentations in my time, probably hundreds over the years. And this is potentially, I think, one of the most important presentations I will give. So I'd like to, start by extending a very warm welcome and my sincere appreciation for you listening to what I have to say this evening. So, trying to move on to the next slide. It's the first technical hitch. There we go. Okay, so this is the layout of my talk. I'm going to start with a brief history of canine genetics research and the Kennel Club Genetics Centre. Many of you are very familiar with us and the work that we do, but I'd like to give everybody the same background so that everyone's coming at this from the same position. I will go through some of the achievements and the resources that the Kennel Club Genetics Centre has developed over the years and our current research programmes. I will then go through details about how the Genetics Centre has been funded in the past and how my ideas for funding it going forwards. So I thought I would start with a little bit of background about me for the people who are not familiar with me. I've worked with many of you for many years. But yeah, I started my canine genetics journey back in 1991, believe it or not, when I had just finished my PhD. I wasn't particularly enamored with science at that point. My PhD had been a bit of an uphill slog. But Jeff Sampson, who many people will know and remember, he had some funding from what was the Guide Dogs for the Blind at that point, and it was joint funding between the University of Leicester, where Jeff was at that time, and the Animal Health Trust. And the role involved identifying some of the very first canine genetic markers. So this is back when virtually nothing was known about the canine genome, and in fact not much more was known about the human genome. In 1995, I moved with my now husband to Seattle to work with Dr. Elaine Ostrander. And my role there was to take these genetic markers, which were sort of these variable positions around the DNA, and put them together to form what's called linkage maps. So this was the sort of first effort to sort of map out the canine genome. And we were using large pedigrees of dogs to do this that had been bred by Gus Aguirre and Greg Ackland, who were ophthalmologists at the University of Pennsylvania. And these pedigrees segregated inherited eye diseases. And so we were able to do a couple of things. We built these genetic maps of the canine genome, but we were also able to map those disease mutations. And this was my first taste, really, of mapping disease mutations. And this paper was published in 1997, and it was mapping of one of those inherited eye diseases, what we know as PRCD these days. And we didn't know at that time that this mutation segregates in, dozens of different breeds. So that was my first, taste of mapping disease genes and sort of realizing the implication of finding these disease mutations and the impact that could have on canine health. In 2001, I came back to the UK with my husband and now my 2-year-old son, and joined the canine genetics group that was at the Animal Health Trust at that time, and that was headed up by Dr. Matthew Binns. And then I started sort of investigating the genetics of canine disease. So I moved from building these resources with which to navigate your way around the canine genome to actually trying to find the mutations that were causing some of those diseases. In 2006, Matthew Binns left the Animal Health Trust and I took over leadership of the canine genetics team and remained there until 2021, when the Animal Health Trust sadly closed, and I moved the Kennel Club Genetics Centre to the University of Cambridge Vet School, which is where we are today. I'd just like to spend a couple of minutes introducing you to the members of the current canine genetics team, because these are the people who really do all the work. I get to give these presentations and to talk about our findings, but these people whose pictures you can see here are the people who really do all that work. So just to sort of go through them in order, Sally Ricketts. Sally was actually doing her PhD at the Animal Health Trust when I joined in 2001, and she leads the idiopathic epilepsy program and the other complex diseases that we are sort of increasingly starting to sort of tease apart. Louise Burmester used to be Louise Downs when she joined. She did her PhD at the Animal Health Trust, investigating progressive retinal atrophy in several breeds and identified 4 different PRA mutations. She now works on lots of different things within our group, including intervertebral disc disease, some of the monogenic diseases that we're looking at. And she really is a sort of key member of the group, working on lots of core projects. Catherine Stanbury really looks after our inherited eye disease program and is usually at any one time juggling about 4 or 5 different projects, looking at different diseases in different breeds. Ellen Schofield is our bioinformatician. She looks after all our huge DNA sequencing files, writes and maintains our bioinformatics pipeline. And again, what she does really goes across all our different projects. Chris Jenkins joined the group in 2014. He completed his PhD just last summer, investigating epilepsy. He works very closely with Sally. He has also worked on some other neurological disorders and has recently been working on histiocytic sarcoma in flat-coated retrievers. Louise Pettit actually joined the Animal Health Trust on the same day I did, January the 2nd, 2001. And I don't know if green fingers applies to lab talent, but Lou is really our very green-fingered molecular geneticist. She works again on many projects and is our go-to person for sort of technical expertise in the lab. Brian will have talked to many of you over the years. He is the first port of call for many inquiries, giving advice on how to get research projects going, collecting samples. If you ask for a swab kit, Brian will send you one and he will collate information regarding that sample when it is returned to us. So he does lots and lots of outreach. And Jim Johnson, who is probably our newest member of the group, joined just a couple of years ago. His role overlaps with Brian to a large extent, but he's involved mainly with collection of epilepsy samples. And again, he works closely with Sally. And then last but not least, James Oliver runs CAGT, which is our in-house commercial canine genetic testing service. And I'll just point out, he is a different James Oliver from the BVAI panelist, James Oliver, with whom we also work very closely. So that can get a little bit confusing to have two James Olivers. But believe it or not, between us, we have 124 years of cumulative and collective experience in the field of dog genetics. And that's 124 years working together at either the Animal Health Trust or the University of Cambridge. So that was quite a surprise to me when I added it up, that we're all that old. It's very nice to have such long-standing and loyal colleagues to work with. So now I'm just going to talk to you, give you a brief history of the Kennel Club Genetics Centre. So as I've explained, canine genetics research did begin at the Animal Health Trust in the early 1990s. But in 2009, this was shortly after the Pedigree Dogs Exposed programme aired, the Kennel Club and the Kennel Club Charitable Trust awarded the Animal Health Trust £750,000 to support the core running costs of the Genetics Centre. And at that point, the research group became the Kennel Club Genetics Centre in recognition of that significant financial contribution. And then, as I've explained, the Kennel Club Genetics Centre moved to the University of Cambridge in early 2021, after the Animal Health Trust had closed permanently in July of 2020. And that move was specifically financed by the Kennel Club Charitable Trust, and it made possible by the University of Cambridge. And I put here, this is sort of our broad manifesto or mission statement, or whatever you want to call it, of our group. You know, the broad aim of everything we do is to identify genetic variants that underpin canine diseases, and to develop tools to prevent, diagnose, and manage those diseases in the pet population. So that underpins absolutely all of our projects. So whistle-stop really of what we have achieved over the years. We are essentially a bunch of scientists. So we have published over 150 peer-reviewed scientific publications. We have amassed a collection of 40,000 different canine DNA samples, about 1 in 3 of which are from dogs that are affected with a condition that we believe is hereditary, or at least has a hereditary component. And we've identified over 30 disease-associated variants that segregate in over 50 different breeds. Inherited eye disease research has sort of been a cornerstone of the group, thanks largely in part to Keith Barnett, who was at the Animal Health Trust and who many of you will remember very fondly, I think. So we have identified more eye disease variants than any other research group in the world. So about 1 in 4 of all the eye disease variants that have been published have been identified by the Kennel Club Genetics Centre. And according to the online Mendelian Inheritance in Animals database, we have actually identified about 9% of all known canine disease variants. So that is significantly greater proportion than we represent of the people working on canine genetics all over the world. We go to a meeting, a scientific conference every couple of years, and there's routinely 250 people at that meeting, of which we represent about 8 or 9 of them. So essentially, we punch above our weight with respect to our productivity, and that is largely due to the sort of coherent team that we have, very complementary expertise that work very, very closely together. I had a lot of fun putting this slide together. This is, I think, every breed that has at least one genetic test as a result of the work that the Kennel Club Genetics Centre has done. So in terms of impact, data from the Kennel Club is about a year old now, so these numbers are slightly out of date. But over 50,000 Kennel Club registered dogs have been screened for at least one variant that the Genetics Centre has identified. And about 384,000 dogs have a known genotype or an inferred genotype. So they've either been DNA tested themselves or they're of known hereditary status. And again, that's according to Kennel Club data from about this time last year. So probably globally, hundreds of thousands of dogs have been screened for our disease variants or the variants that we have identified. And published data shows that dog breeders really do use genetic testing to reduce the frequency of inherited disease variants within breed populations by as much as 90%. So I don't think it's an exaggeration to say that the Kennel Club Genetics Centre's impact on canine health over the years has been profound. So in terms of our current research activities, what are we working on? We continue to develop and maintain that DNA sample data bank that I mentioned, and we share those samples with collaborators in the UK and internationally as well. So if we have samples from dogs with particular disease and collaborators in another country are investigating that disease, then we will gladly share those samples with them. Inherited eye diseases, again, we have an extensive program. We work very closely with the BVA Kennel Club ISDS eye scheme, and we operate the CRIDE database, which is the Consortium to Research Inherited Eye Diseases in Dogs. This is a catalogue of all the known canine eye disease variants, and that is an online searchable tool that anybody can go into and look for information about eye disease mutations. Idiopathic epilepsy, that is looked after by Sally Ricketts, and that is sort of an expanding expanding and developing program. We are starting to investigate the genetics of intervertebral disc disease. This is an opportunity that we've had since we've come to Cambridge, working very closely with Paul Freeman from the neurology team. Intervertebral disc disease affects some of the UK's most popular breeds, and so we're hoping that will give us an opportunity again to have an impact on the welfare of a lot of dogs. Histiocytic sarcoma is another project that we are working with since we've come to Cambridge, so that's an exciting opportunity for us as well. At any time, we are typically investigating multiple monogenic diseases, these sort of classic autosomal recessive diseases, many of which are potentially new or emerging conditions in breeds. Many of our research programs currently involve whole genome sequencing. So when we have sequenced the whole genome of dogs that we're investigating, we place those in the public domain so that they are available for other researchers around the world to use. And we offer a commercial genetic testing service, as I have said, that is known as the CAGT. I found this quote from a presentation that I gave back in 2010, and to be honest, I've probably said more or less the same thing multiple times every year since. And that was that the diseases we study are those that are brought to our attention either by dog breeders or by clinicians, but they have to be debilitating, serious, and or affect large numbers of dogs. And that really is the nub of what we do. We investigate the diseases that are problems for dogs. There's many research groups around the world using the dog as a model for cancer, for example. But we choose the diseases to study because they are a welfare problem for dogs, and that always has been and always will be the reasoning behind the projects that we undergo. So now I'm going to move on and talk about funding. and explain to you how the research that we have done over the years has been funded. So we have benefited from direct funding, probably to the tune of about £1.5 million from many different sources. And I've listed the main ones here, just in alphabetical order, but the American Kennel Club Canine Health Foundation, CAMVets, Dogs Trust, Guide Dogs, Pet Plan Charitable Trust, Waltham Foundation, as well as many breed clubs, societies, and individuals. And that includes our Give a Dog a Genome project that we ran from about 2015. I think it was 77 different breeds contributed £1,000 each to this project. And then with matched funding from the Kennel Club, we sequenced the genome of 89 dogs. Those sequencing data help us literally on a daily basis with all of our disease investigations. And in fact, just a couple of weeks ago, we published a paper describing a mutation responsible for multi-ocular defect in the Old English sheepdog. And these findings arose directly from the Give a Dog a Genome project. But by far our most significant benefactors over the years have been the Kennel Club and the Kennel Club Charitable Trust, who have between them contributed over £3.5 million to the Kennel Club Genetics Centre. And in addition to those overheads, to those direct costs that have come from the Kennel Club, the Charitable Trust, and those additional funders, our overhead costs, which sort of sit in the background, involve the buildings that we sit in, the electricity, the heat, the insurance, any legal advice that we have. And those have been were supported for about 25 years by the Animal Health Trust and are now currently being supported by the University of Cambridge. So this is just a summary, obviously very, very broad figures of our funding over the years, about £3.5 million, as I say, from the Kennel Club and the Charitable Trust. Those other funders, including breed clubs and individuals, comes to about £1.5 million. And then overhead costs, probably also approximately £1.5 million. So canine genetics research has benefited to the tune of about £6.5 million over the years. So the previous funding model has been as follows, that most of our employment costs, and of course our employment costs are the majority of our costs, the majority of those have been supported by funding from the Kennel Club and the Charitable Trust. And a minority of the employment costs have been supported by those additional funders that I listed on my previous slide. The consumable cost, which is the money that the scientists actually spend on DNA sequencing, on test tubes and plastic and other reagents, etc., they have largely been supported by those additional funders. And then the overhead costs or the indirect costs have been supported by those two host institutions. So I've got here an example of the typical costing of a project. So a project that might last, let's say, for a year, will take up about half the time of a researcher who's in charge of that project. There'll be a little bit of my time and some of additional staff time, Brian's time, for example, to collect those samples, and Ellen's time to do the bioinformatics. So the costings might look something like this. So employment costs for a project like that will total something like £40,000. The consumable spend might come to something like £15,000, and then there'll be about £12,000 of overheads on those costs. So that brings, a relatively small scale 12-month project will have a total cost of about £67,000. But that £15,000 of consumables, that's largely what we've had to reach out, or we've typically reached out to those additional funders for. But as you can see from this little table, that is actually a minority of the total costs of all the research that we do. So now I would like to talk to you about funding going into the future and what happens beyond the end of March of this year. So I have here a quote from Mark Beasley, who's the chair of the Kennel Club, and it's in italics because I'm quoting verbatim from Mark here. But he wrote that the health and welfare of dogs is the main priority of the Kennel Club. I'm just going to go and turn my light on, excuse me, it's turned itself off, just a moment. There we go. So plunged into darkness. Yes, I'm sorry. So the health and welfare of dogs is the main priority of the Kennel Club, and it remains committed to investment in canine health research. As announced in February 2023, it is unable to continue to provide general financial support from the Kennel Club Genetics Centre going forward from April 2024, and it will instead be focusing on specific projects, for example, BOAS and IVDD research. So with that information, my team and I have spent a lot of time during 2023 applying for grants from external funding bodies. And I've summarised here the applications that we have written. So we have put 3 applications into the Pet Plan Charitable Trust, 2 applications into Dogs Trust, 2 applications to CamVet, and 1 application to the Waltham Foundation, 1 to the BSAVA, 1 to the Activities Health and Welfare subgroup, that's the Kennel Club subgroup, and 1 to the Kennel Club Charitable Trust for salaries that were not covered by the grants listed above it. And if those applications had all been funded, that would have come to a total award of £970,000. For those of you not familiar with the funding application process, I've just outlined it here. So these funding bodies, such as the Pet Plan Charitable Trust, typically put a call out once a year where they invite research applications. So people such as myself will submit usually a preliminary application, and then if that is successful, we will be invited to submit a full application. Sometimes there's just 1 application. Those applications are then reviewed, usually sent out to peer review, and then some months later we will get a funding decision. That whole process takes about a 12-month cycle. So we are restricted usually to applying for these applications once a year. We can't just get in touch with Pet Plan or Dogs Trust and ask them to consider an application. And writing each application, that in itself is a pretty time-consuming process. And then, of course, not all the applications are successful. So this is the outcomes of those applications that we wrote. So of the 3 applications we submitted to Pet Plan, 2 were successful, 1 was not successful. Our 2 applications to CamVet were successful, as was our very small application to the Kennel Club subgroup. Unfortunately, our application to the Kennel Club Charitable Trust for that fill-in salary costs was not successful. The Charitable Trust, I think, remains very supportive of the Genetics Centre, but was not, unfortunately, able to fund us on this occasion. Unfortunately, neither of our Dogs Trust applications were successful, or our application to Waltham, and our BSAVA application is still under review. So our successful applications have brought in an income of just under £222,000. But that only covers less than about 18% of our employment costs for the 12-month period ending at the end of March of this year. So there will be a significant funding gap beyond April of this year. And essentially, a new funding model is required, and funds are urgently required if the Canine Genetics Centre is to continue to function in its current format beyond the end of March of this year. So I think it is time that we have to face reality, and it's time, I think, to pass the funding baton to the stakeholders. And I use that term stakeholders a lot. So I did actually look up the definition of stakeholder to make sure I was using it correctly. And a stakeholder is a person with an interest or concern in something, especially a business. So I think in this context, what I mean by stakeholders is are the dog breeders, the dog owners, and what I'm going to call the breed custodians, those of you who have particular breeds that you're very attached to and whose future is all important to you. And I think ultimately this model could provide a more stable funding structure than relying on a relatively small number of funders, and could, I think, potentially provide stakeholders with greater agency over the research that we do. And I hope that the cost to individual stakeholders, will be relatively small if that cost is shared widely. And we all know, effort is decreased if it can be shared. So that's I hope the model that we can employ going forwards. So I know the question on everyone's lips is, how big is that funding gap? So to retain all the Canine Genetic Centre team for a 12-month period beyond the end of March will require an additional £345,000. There are over 200 breed communities in the UK, probably closer to 230 if we count non-Kennel Club registered breeds. And we are here for all dogs. We do not need a breed to be a Kennel Club registered breed for us to work with them if they have an inherited disease problem. If each of those breed groups could contribute about £1,500, we would be on a financially stable footing for the next 12 months. And I am, very, very aware that £1,500 is a lot of money, but it's not so much money if that is shared between clubs, between societies, and between individuals. And even for the numerically smallest breeds, it represents, less than £10 per registered puppy, and it's the equivalent of about £1 for every Kennel Club registered puppy, or every puppy that was registered with the Kennel Club in 2022. I am not insinuating that cost should be added to the price of a puppy or anything like that. I'm just using that as a ballpark figure for sort of an approximate costs if the costs can be shared widely enough. Myself and my team, we will continue to apply for grants and try to identify alternative sponsors and have conversations along those lines. But those conversations will take time, and their funds are needed urgently to fill this funding gap. And realistically, they're needed before the end of February if all the staff are to be retained. I expect many people will be thinking or will want to ask, if I make a donation, how will my breed benefit? So I'm just going to remind you, I hope, of some of the things that we're doing that will benefit your breed. So we will continue to maintain that bank of currently 40,000 DNA samples, but that will be maintained and developed, and that will include archiving for currently something like 12 breeds that we offer that service for. Those ongoing research programs that we have and that we are trying to develop, so inherited eye diseases, idiopathic epilepsy, intervertebral disc disease, cumulatively those are relevant to a large number of breeds. We are really expanding our use of whole genome sequencing to identify novel and emerging monogenic disease mutations. And we're really in a position now to identify those mutations almost as soon as those first affected dogs are born. And then just a reminder that all the whole genome sequencing that we do, we place that data in an anonymised fashion in the public domain for researchers around the world to use. And just as a reference, in 2023, we sequenced the genomes of 81 dogs of 22 different breeds, and that information was all shared with other researchers. And I'm just going to show you this slide again. You know, this is what you will be contributing to, this team of very dedicated and very talented people with all their cumulative and collective experience. I'll just point out that the funds that I am requesting will not need to be used to support the salary of James Oliver. The Canine Genetic Testing Service is self-supporting. So how can you contribute? You can go to our website and make a donation. So if this is the address of our website, but if you put the Kennel Club Genetics Centre into Google, you should find us. And then up at the top right is a donate button. If you press that, you'll be taken to this page. So this is organised for us by the University of Cambridge, and you can have the opportunity to make a donation, either a one-off donation or a recurring donation, and you can donate as little as £10. The funds that are donated in this way will go into the University of Cambridge bank account, will be held safely there, but they will be ring-fenced for canine genetic research. Other ways in which you can help, if you would prefer to make a bank transfer or send a cheque, we can accommodate that as well. And if you email donate@caninegenetics.org.uk, we will reply and tell you how to send us a cheque or make a bank transfer. As soon as possible after this presentation, hopefully by tomorrow morning, we will email you a link to this recording. Please share that. Please share that with your family, your friends, your club members, your personal network, using whatever your preferred mechanism is. We would really like to encourage regular donations. That will give us an increased stability. So please give us feedback. Tell us how you think we could thank stakeholders who make regular donations. That might be through particular research talks, workshops, research updates and newsletters, specific research projects, discounted genetic testing. We are really open to ideas. One idea that we think might encourage people to make donations is if we set up an advisory panel of independent people who can add some, help at some spending accountability to the stakeholders. And if you have any questions at all about this that are not, maybe you're not getting the opportunity to ask today, please email me. This is the email address that I'm using for these inquiries. Many of you will have another email address for me, which also works, but if you email this, Catherine@caninegenetics.org.uk, I will make sure that all those questions will be in the same place, and I'll be sure to see them. This is just my penultimate slide, some acknowledgements. So on the left are the research team, past and present. So all the work that I've talked about, all the achievements that the Genetics Centre has made over the years have been due to the work of all these people listed here. And then I would like to thank the funding bodies that I've mentioned before during my talk. And just to make it clear, these are organisations that have funded us in the past. They are not necessarily all funding us now. I'd like to thank the breeds, clubs and societies and individuals who have funded us and have also contributed DNA and information about their dogs. We wouldn't be able to do anything without any of that. And finally, I'd like to say a really big thank you to everyone who have sent messages of support over the last few days. I haven't had time to reply to everybody, but I have read them all, and they have all been hugely, hugely appreciated. And I will just leave you with this slide. This is a link to a talk that Cambridge University wrote about the group. There's a QR code at the bottom if you would like to open up that story and read it. And for now, I'll just leave you with the picture of this beautiful Kaya, and invite you to send us some questions if you would like. You can do that by typing your question into the Q&A section. And I think to give people time to do that, it looks like we have got some questions coming in already. I think maybe we will break. We started a little bit late, a few minutes late due to some technical hitches, but we will maybe resume back here at about 5 to 8 to give you time to type your questions in, go and have a cup of tea or a glass of wine. We'll resume at about 5 to 8, and then we will answer as many of those questions we can over the course of the next sort of 10 or 15 minutes. I will remind you that if you cannot listen to all of those, we will be obviously recording, and you can catch up with those tomorrow. But for now, I'm just going to mute myself and turn off my video and be back with you at about 5 to 8. Thank you very much. Okay, so I hope you can hear me again. That I've come back online and everyone can see and hear me again. We are receiving a lot of questions, and that doesn't surprise me at all. I think we've received a lot more than we are going to meaningfully be able to answer in the next 10 minutes or so, to avoid this overrunning too much. We will take every question and copy it and answer it specifically on our website, and we'll send you a link to all those questions as soon as we can. But there are some recurring themes, so I will just, in the next 10 minutes or so, go through a few of those that are coming up more than once. So there are people asking about the short time frame. Why have I left it so late? And I do totally recognise that breed clubs will have to have conversations about making donations. And the truth of it is, I did not realise that there was going to be such a large funding gap until very recently. We only heard back from the result of our applications to the Dogs Trust a couple of weeks ago, and I only heard back regarding our quite large application to the Kennel Club Charitable Trust towards the end of last year. So that is why we haven't, unfortunately, been able to give people more notice of this funding gap. There's quite a few people asking whether this is going to be an ongoing requirement from breed clubs. I think that depends on whether we can attract other alternative sponsors to basically step into the Kennel Club shoes. I will be trying very hard to find alternative sponsors, but it's likely that I think, yes, that there could be an ongoing requirement for you as stakeholders, be it, the breed clubs or individual breeders, to contribute towards cost. I would prefer for that not to be the case, but as I said, I also think that maybe having a broader base of funding will give us some degree of stability. So I can't be sure, but my prediction is that there may be an ongoing requirement for stakeholder contributions. A lot of people are asking questions about their breed-specific projects and whether they will continue. I hope so, and that's why I'm trying very hard to keep us going. But the truth is, if I can't attract enough funding to retain the members of the team, then that will have inevitable consequences for some of the projects. But it is absolutely our intention to keep as many of those projects going as we possibly can. We've had a question about obviously some breed clubs contributed funds to us when we were at the Animal Health Trust, and those funds were sadly lost. So we've had a couple of questions about the stability of the pot if people make donations going forward from today. All the donations that you give via our website, they will go into a University of Cambridge account and will be looked after by the University of Cambridge. And given the University of Cambridge has been around for 800 years, I don't think it's going anywhere anytime soon. So I think, yeah, you can have a large degree of confidence that your donations will be safe, and they will be ring-fenced for canine genetics research. In the event that the genetic centre doesn't sadly survive, then they will be used for the benefit of dog welfare, and I have the guarantee of my head of department for that. We have somebody had posted a question about Crufts, whether we will be at Crufts and whether that is an opportunity for fundraising. We will have a stand at Crufts. That has been arranged, and so I will be there with the CAGT, the canine genetic commercial genetic testing service staff, selling DNA tests, and I will be there, and I would certainly encourage anybody to come and talk to me about the funding situation then. But bearing in mind that Crufts is the beginning of March, and essentially our employment contracts end at the end of March, I would really put a plea out for those of you who can make donations prior to that date to please do so. I have this question is coming up a lot, and I don't think I can answer this, but the question about I made reference to the funding gap spread between, it would be the equivalent of a pound for every Kennel Club registered puppy. We've had a lot of questions asking about that. Why doesn't the Kennel Club add a pound to the price of a puppy registration? I do not work from the Kennel Club, and I am not in a position to answer that question, I'm afraid. So I will have to defer that to somebody from the Kennel Club who may wish to send us an answer. I've had a question about the Give a Dog a Genome project that I referred to, that was a crowd-funded, stakeholder-funded project from about 2015. I think we started that project. Somebody was asking, had that project stalled? It seemed to have stalled. We are no longer sequencing genomes funded by the Give a Dog a Genome project, but every single genome that we sequenced is used on a daily basis almost for all of our studies, either specific projects aimed at the breeds of those genomes or as controls for projects in other breeds. So they are absolutely useful, and those data are in the public domain and will remain so indefinitely. And so those genome sequences will be used forever for the benefit of dogs. So although we are no longer sequencing dogs as a part of that project, we still sequence genomes of dogs very, very regularly. The Give a Dog a Genome project is no longer recruiting samples, but the data are very much actively used. And then I think finally, people are asking about ideas for attracting commercial funding. We had somebody who was an anonymous post actually, but offering their services to help attract commercial funding. I would very much welcome any suggestions from anybody, either in a position to offer potentially commercial funding or to help us attract or with ideas for how best to do that. None of myself or none of the other people in my group come from that background. We are scientists, we're geneticists. So I would certainly be very grateful for any help in that direction. And then I think one final comment. So somebody's asking about our list of projects. We do have a website. We are conscious that probably hasn't had as much TLC as it could have done in recent years, but we intend to keep that updated. But yeah, there are sort of a list of our broad research projects. So yes, please look at our website. We've had one question about our CAGT, our genetic testing. What happens to that? So all the profits from CAGT, once we have paid our costs, do come back to the Genetic Centre to help fund research. So that is one other thing that you can do to help, is to use CAGT for your DNA testing needs, any profits from above our costs will come back to the research centre. If there is a genetic test that your breed uses or makes use of that CAGT does not offer, then please talk to us. We're a very friendly bunch. James, who runs that, would happily have a conversation about adding a genetic test to us, and that is another way in which you can indirectly help our research income. And I think that is probably as many questions as there's one final. If all the breed clubs all agreed to donate each, would we be advised when you've reached the cutoff amount? We will absolutely be keeping you updated. We will post regular updates on the website and also email them out to all the people who have registered for this talk tonight, telling us where we're going. So we'd very much like to keep you in the loop. And now I can see it's 8:10. I am conscious that I have kept everybody a little bit past when we said we were going to finish this talk. So I think we're going to leave it there now. I will, as I say, we will go through all the many other questions and post our answers on our website, and we will also share a link to this recording that you please feel at liberty to share that with your networks, whoever happen to be in your networks. So once again, thank you very, very much for giving up time on a Tuesday evening to listen to my talk. I really do appreciate it. So good night, and have a very good rest of your evenings. Thank you.